Right, welcome back. Um, we've talked about free nerve endings and we have talked about chemosensory structures. I want to shift gears now and talk about vision. And vision is going to involve what we call photoreceptors. And in a photoreceptors rely on chemicals that will change shape when exposed to certain wavelengths of light. And we'll talk about how we have photoreceptors in the back of the eye, and we have this very complicated structure, like I have drawn on the board here, that represents the various parts of the eye and their role that they play in projecting the light energy onto the retina. The retina is where we have the photoreceptors. So when we look at the eye, I've got a little picture up here. You can see that we have, at one end, we have the cornea, and that's the clear outer surface of your eye, and that's what light passes through first. And behind the cornea and in front of your lens, we have the anterior cavity. I have up there chamber, but it's really cavity. So it's the anterior cavity, and the anterior cavity is divided into anterior chambers and posterior chambers. And we had said that in lab, we had said that the anterior chamber is in front of the iris, but behind the cornea, and behind the iris, but in front of the lens is the posterior chamber. That's all part of the anterior cavity. And this cavity is filled with aqueous humor. This is a fluid that is produced by the ciliary body, which is what is present right there next to your lens, and it is what produces the intraocular pressure in your eye. You may have heard of glaucoma. Glaucoma is having high pressure in your eye. It results from having excess aqueous humor, which is produced and is found in this portion of your eye. What does the iris do? So let's think about the functionality of the eye. The iris has both smooth and skeletal muscles which are able to make the pupil, which is a hole, make the pupil large or small by either dilating your eye, your iris, or constricting your iris to make the opening smaller. And so what it does is that it controls the amount of light that is coming into your eye. The light is then passed through a lens. What does the lens do? The lens has ligaments, suspensory ligaments that are holding on to the sides of it, and it's going to be pulled. And the reason it gets pulled on is so that it changes shape, and it is going to focus the image, focus the light on a very specific location on your retina, which is in the posterior cavity. And the posterior cavity, of course, is filled with vitreous humor. Now, unlike the aqueous humor, the vitreous humor you've had since embryological development. So you have the same vitreous humor throughout life, whereas you're constantly producing the aqueous humor. In the posterior cavity, we have the retina. The retina is this thin piece of tissue lying against the back of the posterior cavity, and it is held in place by the vitreous humor. That is the job of the vitreous humor. It's to keep the retina held in place. And we had said the retina is going to be the site where you are going to sense the light energy that has passed into your eye through the lens and is focused on a specific location in the back of your eye. And the photoreceptors are going to be connected to bipolar neurons, which are going to pass through the optic nerve to your brain. Okay, so this is the physical structure of the eye. All right. So what I've drawn on the board, I've changed the picture on my board to represent what you see with a cross section through the retina. And up here you notice we have the light path. The light path is coming, and this is the back of the eye over here. And you notice that the light has to go past the ganglia, the bipolar neuron, and finally to the photoreceptor. So light, this light here, we had said, where did it come from? It came from the lens, which had come through the pupil, which had come through the cornea. So it came from the front of the eye, through all of these structures, it's passing through the vitreous humor, and it finally reaches the retina. And what does it do? It has to go past the ganglia. It has to go past what we call the bipolar neuron, and eventually it will reach the photoreceptor. And we have two different kinds of photoreceptors. We'll be talking about rods and cones. But we have, when the light hits the photoreceptor, we'll talk about a chemical change that occurs, which causes the bipolar neuron to send a signal to the ganglia through the optic nerve to your brain. So it's not, the funny thing about oftentimes the funny thing about the way the human body is designed, it's not designed like an engineer would design it. No, far from it. The light has to pass through past these other structures to get to the photoreceptor. So that's a unique, strange design quality to how the retina is designed. All right, I've drawn another picture. You know how much I like pictures. Okay, so again, we're going to be concentrating on the retina. We're going to be concentrating on the retina. But when we talk about critical vision, we're going to talk about light. We're going to talk about light coming in, we'd said passing across the cornea through the lens. And the lens is going to aim the light, focus the light on a very specific location in the retina. And that is a region called the fovea centralis. The fovea centralis is the site where you're going to have critical vision. Now, what is critical vision? This is where, like when you're reading or you needing to see things in extreme detail. Now, I had said just a minute ago, I had said that we have two different kinds of photoreceptors. We have what we call rods, and we have cones. The cones are concentrated in the fovea centralis. And the cones are what are going to allow us to see in bright light and to see things very clearly. Now, you notice we have just this one spot where we have all these cones, and we have this really large retina. And throughout that retina, we have photoreceptors, but the majority of those photoreceptors are going to be rods. Now, rods are able to see in much dimmer light. As a matter of fact, you can't see, they are non-functional in bright light. They're only functional in dim light. Similarly, you have to have a lot of light to activate cone cells. So we have these two systems that are going to allow you to see in dim light, but not very clearly, and in bright light, and to see things in extreme detail. And so, like when you're reading or watching this video, the light is being focused on the fovea centralis, and that is then sending its signal through the optic nerve to your brain. All right, so I was just mentioning about the different types of receptors. We had said that we have cone receptors, which allow you to have very critical vision, to see things clearly. And then we have rod receptors, which allow you to see in very dim light, but you don't see much detail. It's very hard to read in the dark. That's why. So to illustrate this point, if we look at the flow of information that I mentioned before, I'd said the light passes to the photoreceptor, and the photoreceptor sends a signal to a bipolar neuron, which passes it to a ganglion, which then sends it on to your brain. When we talk about the cone receptors, every single receptor has its own neuron. Every cone receptor has its own bipolar neuron. Now, in contrast to that, with the rods, the rod receptors share bipolar neurons, as many as 600 receptors per bipolar neuron. Now, why would that matter? So if you think about your television, this is how your eye sees. It's the exact same thing. So when you look at your television and you see an image, it's a bunch of dots. And every one of those dots is a different color, and it puts together a composite image that you see, well, like me, my lovely face, coming to you now. So it puts together a composite image. Now, in your cones, every one of these receptors is putting out a pixel. It's a signal that's like a pixel that's put together as an image that you see. Now, when we talk about the rods, you have up to 600 of these pixels, essentially, of these receptors that are feeding into a single pixel. So 600 receptors feeding into a single pixel. So everything's blurry as a result of that. But they're able to collect a lot of light, so you're able to see in very low light conditions. So we have advantages to both of them. You can see color with the cones. That we'll talk about how that works in a minute. And you also can see detail, but you can't see well in very low light conditions. All right, let's talk for a minute about photoreceptors. Now, photoreceptors are going to contain a chemical which is called a photopigment. And a photopigment, let me change my angle of my view here. A photopigment, these are chemicals that are going to change their shape when they absorb light energy. And chemicals can take different forms. They call it the cis form and the trans form. In the case of the photochemical that we're going to be talking about, it's something called rhodopsin. This is the chemical that is found in the rods, and they're very closely related chemicals that will be found in the cone cells. So what's going to happen is rhodopsin is going to change shape, and it's this shape change that is going to result in the sending of a signal through the bipolar neuron, through the ganglion, back to the brain. Okay, I want to make sure everybody looks at your notes, the PowerPoint notes. I've drawn something on the board that is representative of these chemical shapes, but I didn't put all the detail in. If you look at this, now what happens when we talk about light hitting rhodopsin? We had said that the rhodopsin and retinol A, this retinol, this is the part of retinol that changes shape. It's something called rhodopsin. And so rhodopsin, when light hits it, it's going to change shapes. So if you notice down here, I say no light. If there's no light, the rhodopsin has this bent form, and this bent form is called the cis form. Now, when it absorbs light, this bend in the shape of the molecule, it disappears. It straightens out. And when it straightens out, this is called the trans form. And this is the change that happens to the photoreceptor that causes the signal to be sent to your brain, saying, I have light. You know, this pixel in the TV picture, if you will. So we had said the way you see is kind of like a television. It's very similar to a TV. And so you have this composite picture, and you either have light or you don't have light, and you say, oh, it's a puppy. or whatever you're looking at. And so it's this change in the form that is going to result in the signal being sent to your brain saying that the photoreceptor has absorbed the light. Okay, so we had said that we have these two different forms that the rhodopsin can take. And we had said the cis form is bent and the trans form is straight. Now, when there is no light, in the absence of light, the rhodopsin is in the cis form. That's the bent form. Now, when it is in the cis form, the rod cell is going to be secreting glutamate onto the bipolar neuron. Glutamate is an inhibitor of action potential by the bipolar neuron. So in the absence of light, the bipolar neuron doesn't send a signal to the brain saying we have light. When you have light, as we see down here in the lower left, we had said that converts the rhodopsin to the other form, that straight form, the trans form. And when you do that, it stops, the rod cell stops secreting glutamate onto the bipolar neuron. So the inhibition is removed and a signal is sent to your brain saying, I see light. That's how it is. That's how it works. That's how you're able to put together this composite picture that you see as an image with vision. Color vision. All right, so now we understand when the photoreceptor responds, absorbs light energy and changes shape, it's going to send a signal. Now, so rods are simple. They either say we have light or we don't have light. And any of the visible light spectrum light will cause this transformation from the cis form to the trans form. Now, with the cone cells, as I'm sure all of you know, we have color vision. So how does that work? How on earth can you distinguish colors? Well, it's by the receptors. We have 3 different kinds of cone receptors. And what that means is each of these different types or categories of cone receptors, they're only going to respond to a small portion of the visible light colored, visible light spectrum represented by the different colors. And we have 3 different colors of light, and those are different wavelengths of light that are represented here. One type of cone receptor is going to respond to yellow-orange light. Another type is going to only respond to the green light spectrum. And the third type is only going to respond to the blue light spectrum. And so by combining these, and your brain knows that if it receives a signal from a photoreceptor that responds to green light, you perceive that as green. Your brain says, oh, that must be green. So we have these different kinds of cone receptors that are going to produce color vision. All right, our final point dealing with vision is going to have to do with this duplicity theory. And the duplicity theory basically says that no single receptor type is able to provide us with all the forms of vision that we have. In low light conditions, we don't have enough light for the rods for the cone cells to be functional. In low light conditions, we don't have enough light for the cone cells to be functional, so the rod cells are providing us with the vision we have, when we're walking around in the dark. And then when you have bright light, how does a black and white TV work? Let's think about that for a second. A black and white TV has various shades of black and white. And you get like these black and white images because some of the pixels are black and some of the pixels are white. If you're in bright light, every single pixel would be white. So you're not going to see anything. That's the way the rod cells work. So when you're in bright light, the rod cells are of no value because they're all in the transform. They're all saying, yes, we have light. Now, so when we're in bright light, the cone cells take over. The cone cells require more light, so they don't function in dark conditions. But if you have a lot of light, then what they can do is you can see very clearly, because each one of them has its own bipolar neuron, so you have a very clear, crisp picture. And also, you can see the different colors. So this gives us the clear critical vision and color vision, because your brain knows that those individual cones are only going to send a signal if they're seeing the yellow-orange light, or if they're seeing the green light, or if they are responding to blue light. So this is called the duplicity theory of vision. And this is the end of our discussion on the photoreceptors and vision. We have one more section. We're going to talk about hearing and equilibrium.